So in the dating game, the male mosquitoes are mostly losers.
Hello, everyone, and welcome back to People, Parasites, and Plagues, a podcast aimed at delivering information about the fascinating pathogens among us from the scientists who study them. I am Kim Klonowski.
And I am David Peterson, your host for today's episode. Today we will learn whether the mosquito's foot is malaria's Achilles heel.
Our guest today is Dr. Doug Paton, faculty in the Department of Infectious Diseases and a member of the Center for Tropical and Emerging Global Diseases. Doug, welcome to the podcast.
Hello.
So, Doug, one thing that we really enjoy talking with folks about is their pathway to science. So start anywhere you want in your personal timeline and let us know how you arrived here.
So it's a pretty circuitous path. I'm originally from Dundee in Scotland, and I went to the University of Dundee for undergraduate. I studied microbiology. I didn't enjoy it very much, but in my final year, I had a class on molecular parasitology with Alan Fairlamb, who's a certainly very big researcher in the trypanosomiasis space. Did a lot of work on new drugs for treating that as a neglected tropical disease. And he really I make the joke that he kind of radicalized me a little bit. He was always a little bit up in arms about the state of drugs for that particular disease. It's very it's considered a neglected tropical disease still, although the cases are going right down now. But at the time, this is 2003, there was a problem where there were drugs available, but they were only being manufactured for use in cream to reduce hair growth in women, and they weren't being made available for trypanosomiasis. So he was really angry about that, and it came across in his lectures, and I got a bit grumpy about that too, and this kind of launched me on the trajectory I ended up on. I didn't do particularly well in my undergraduate, so I ended up having to work in the British equivalent of the IRS for 3 years to kind of put together money to get to do a master's, but eventually got to the Liverpool School of Tropical Medicine and did a master's in vector biology and parasite disease control. That went really well, and that kind of led to a PhD at the University of Kiel, which is a really small university in the English countryside, but had a really great vector biology department. So I studied the sort of behavioral genetics of Anopheles gambiae. That's the vector of malaria, one of the biggest vectors of malaria in sub-Saharan Africa. And then, so that was 5 years there, and then I did a lot of field work as part of that PhD. I worked in Mali, I worked in Burkina Faso, and right at the end of my PhD, I was doing field work in Burkina Faso and actually got malaria while I was there and was hospitalized.
I am so jealous.
Yeah. Well, you are our second guest who's had malaria.
Oh, dear.
Yes.
So this is kind of shameful because I obviously had prophylaxis and was supposed to be taking it. I started it a day late. I got sick 8 days after arriving, which suggests I got bitten pretty much immediately after leaving the airport, and just when I was in this kind of window of vulnerability. And so in that situation, because I was naive, they treated me like a complicated malaria patient. So I was hospitalized. They gave me intravenous quinine, which is very, very unpleasant. It's very, very toxic drug. So I was completely out of it. And that's where I met my to-be postdoc advisor. So I was out there with some colleagues from Harvard University that were studying the same mosquito populations, and their boss, Flaminia Catarucci, was out with them while I was in hospital. And sort of as I got out, we went out for dinner together, and that was when I met Flam.
So let me just stop you for a second. So how did they connect with you in the hospital? Like, was this like, oh, there's this?
Oh, no, I'd been working with them.
Oh, okay. You had been working with them.
We were in the same guest house. We were kind of overlapping.
Okay. So they knew who you were before. It wasn't like we came into the hospital and we found this poor guy who got malaria. Let's get him to study malaria.
They all came down with malaria.
Okay.
We're in the same ward.
Well, yeah, no, we'd been working together at same field sites, everything like that. So shared resources in the lab, and like we hit it off pretty quickly. It was actually really great. And then, I just finished my PhD. I just graduated and was looking for jobs and spoke to Flaminia about it. And she had at the time, she had two labs. It's kind of complicated. She had a lab in Perugia, Italy, and one in her main lab at Harvard. And she had a postdoc opportunity coming up in Italy. So I kind of upsticked from the UK and moved to Italy for 2 years to work with her there. That lab eventually, my understanding, and this may not be completely true, so dose assaults, but my understanding is that the Italian administrators of the university basically said to Flaminia, you can either be a professor here or you can be a professor at Harvard, and we don't want to continue this kind of hybrid approach where you have two labs. And she chose to kind of stick to Harvard. We just got a little money from the Gates Foundation at that point, and so that was enough to kind of justify me joining her as a full-time postdoc at the School of Public Health at Harvard. I spent 8 years there as a postdoc, which is a great time. And then, yeah, just that went pretty well, especially the latter half. Thankfully, not the first half, but the latter half, good timing. And then I started at the University of Georgia as an assistant professor 2 years ago, almost exactly 2 years ago now.
So when you, as a grad student, first went to Burkina Faso, was that your first time in Africa?
So I'd been to Mali twice before. So I did 2 field seasons in Mali in 2010 and 2009, 2010. And then we had been working there. Mali is an area that's certainly the southern half of Mali, which is the Sahelian region, has like year-round rice cultivation. And so it's very, very intense malaria transmission associated with the mosquito populations there. And so it's been a great place to study malaria. There's very high intense transmission. There's a lot of insecticide resistance in the mosquitoes. But that was also the time of the Libyan civil war, and there had been some destabilization in the region. And so we couldn't go back to Mali because it had become kind of less safe for us. So we started going to Burkina instead. So yeah, that was my first time in Burkina for sure. But I'd spent quite a lot of time in Bamako prior to that. So
Yeah, we talked off mic about, because I also spent some time in Mali, in Bamako, and I just remember being blown away when I first got there because things seemed so different. But then at the same time, there's a lot of things you recognize. There's stores, there's roads and cars, and I guess my brain was a little confused between the two extremes.
yeah, it's definitely an experience. I really cherish it as someone who works in this space in the malaria field. It was really an amazing experience to have, especially that early in my career. It really kind of contextualized a lot of the work we do and the kind of people that we're trying to help by trying to interrupt this disease as well.
So as a non-parasitologist here, I do like to try to bring it back sometimes to people who maybe aren't thinking about parasites and how these things are transmitted. And we talked about, rice cultivation, right? So we think about maybe in our own homes and our gardens, we don't want standing water. Well, when you're talking about rice, this is standing water, right? So these are the prime places to study malaria. So it was a really unique experience, I imagine.
Yeah, that's really it. Those areas around some of the larger river systems and irrigation systems in that particular part of the Sahel, which is subtropical, so it has high seasonality, right? So you get a lot of malaria in the wet season and typically no malaria or very little malaria in the dry season. But where you have year-round rice cultivation, you kind of buck that trend and you get constant transmission all year round. You also get a lot of insecticides leaching into the groundwater from that rice cultivation, which leads to problems in the mosquito populations too, not only through use of directed insecticides against those mosquitoes, but just through environmental exposure. So it's really and it's, if looking at the maps of transmission, it's always been one of the most intense areas of very, very high prevalence in children under the age of 10. It's also one of the most economically suppressed areas of the world as well. So there's a lot of poverty and inequality there as well. So all of these kind of factors combine to make it one of the most intense malaria areas. And as a result, kind of if you're going to design a new intervention, it's the place you want to go and test it. So.
Right. And we've had quite a few guests who have had some work at some point or have worked with collaborators in Mali. So it all really makes sense. So why don't you tell us about your current research question?
So I'm interested in mosquitoes, if that wasn't already clear, right? So, I've done a lot of work on mosquitoes actually, and that's kind of morphed over time into being more interested in the malaria parasite and more interested in how they transmit parasites. So again, most of my background is in mosquito behavior and genetics, but increasingly we mostly work on the parasite itself. So malaria parasites to transmit between one human and another have to go through a mosquito. It's a completely obligate step. And the mosquito isn't kind of just carrying the parasite from one person to the next. It's actually being infected by that parasite. There's a complex interaction between the parasite and the mosquito as it's kind of developing. It takes a long time, about 2 weeks in the lab, a little bit shorter in the field for a parasite to get into a mosquito and then out the other end. However, they're pretty difficult to study, or it has been historically harder to study that stage, especially under lab conditions. And so the biology of parasites as they transmit through mosquitoes is relatively poorly understood. But kind of main, I guess the headline result of my postdoc was that we discovered that we can use small molecules, like basically anti-malarial drugs, we can give them to mosquitoes, and that can kill parasites as they transmit through the mosquito. And that's important because our insecticides, which we currently use to control mosquito populations, are no longer working as well, especially in those areas of heavy agricultural use of insecticides. So we're in a situation now in Western Burkina Faso and Southern Mali where insecticides just don't work against those mosquitoes. And there's a very limited number of insecticides you can use in bed nets. The primary way we prevent malaria transmission in those areas is through the use of insecticide-treated bed nets. Those are just a classic bed net, but it's got impregnated fibers that have drugs in them that kills the mosquitoes on contact. And they have to be safe for close human contact. So you have children sleeping under these, they have to be very safe. And so there's only one class of insecticides that are suitable for this use case. And now we have really, really high grade resistance throughout most mosquito populations in sub-Saharan Africa to that particular drug. So we were thinking about this problem. So, if we can't kill them, it's a slightly hand-wavy conversation I was having with Flamini at the time. We had many of those. One of my great pleasures as a postdoc. But we were kind of asked the question, what if we could cure the mosquito instead? If we can't kill the mosquito, can we use a drug to cure it instead? Would that be worth doing? Is the first question we asked. One was, could we do it? And the other was, is it worth doing at all? So we did some modeling. Actually, we were working with an epidemiologist, Caroline Buckey, also at Harvard, who had built some models so we could kind of in silico sort of test interventions a priori sort of before we started, and it turned out that if we kind of modeled out the idea of kind of curing or preventing transmission of parasites to mosquitoes, it could actually have a pretty big effect. So that was the kind of, is it worth doing? The next question was, can we do it? And that was a little bit more up in the air. But so I what we were able to show ultimately is if you take drugs, particularly the drug atovaquone, which is the primary component of Malaria. If you've ever been to a malaria endemic country, that's the drug you'll typically be prescribed. It's very safe. You have to take it every day. And actually, it turns out atovaquone is not a very good drug. That's why you have to take it every day, because it doesn't stick, it doesn't absorb. It's very, very lipid soluble and very insoluble in water. And so it's not absorbed by your body, but it is absorbed in oils. That's why you have to take it with food. It turns out it's a terrible drug for giving to people orally, but it turns out it's excellent for letting for mosquitoes to absorb it through their legs. This kind of gets to your point earlier, David.
Yes.
So mosquitoes are covered in wax. Their outer layer is wax, so they don't dry out, and so they can repel water if they get rained on. And when they land on surfaces, they can absorb compounds into that waxy layer that then gets into their internal biology. So atovaquone in particular, as it turned out, actually is one of the best drugs. I could have started with this. I tested this drug through tarsal contact, we call it. So the tarsi are the lower part of the mosquito's legs. We let them land on a surface with it covered in atovaquone, and they absorb the drug, as it turns out. And that's enough to prevent them from being infected with malaria for 2 or 3 days. So that led to a sort of flurry of activity. This was a very unexpected result. Again, it came out of a very hand-wavy conversation. I had no idea whether this was going to work, but it turns out so we can treat mosquitoes with the drug through tarsal contact. And the tarsal contact's the important part, because we can feed mosquitoes drugs. We can feed them sugar with drugs in them. But the idea of them being able to just passively absorb a drug from a surface was kind of where the novelty was. That it kind of makes it compatible with the idea of putting an anti-malarial drug in a bed net, for example. And so that research is kind of, the latter half of my postdoc was kind of figuring this out. We did a couple of screens to find additional compounds that work that way. It turns out, this was the interesting part, I think, is that the only compounds that work through tarsal contact are mitochondrial inhibitors. So the parasite is very vulnerable to mitochondrial inhibition through the mosquito. That had been shown a couple of times in different ways, but it seemed to kind of really emerge from our work is that the compounds that work best were all inhibitors of parasite mitochondrial function, so energy metabolism mostly, although we don't know, and I'll get to that. So all the compounds that worked were mitochondrial inhibitors. Something about that target, so the drugs specifically target the complex 3, which is the central part of the electron transport chain. Something about that target, which is inside a lipid bilayer, so it's in a very hydrophobic pocket inside a hydrophobic pocket, seems to make those drugs suitable for passing through the cuticle of the mosquito. That's a process we don't fully understand, and actually it's one of our research subjects. It's like what makes a compound good at being absorbed by mosquitoes. You'd think the insecticide industry would have figured this out many years ago, but either they have and they haven't shared the data, or if the data doesn't exist. So we're having to start a bit de novo there. And so the kind of second part of that, and actually having buried the lead somewhat, the major part of my research is, again, we really found most of the compounds that work were malaria parasites, as they were infecting the mosquito, were mitochondrial. And we've known for a while that the mitochondria was a kind of vulnerability of the parasite as it goes through the mosquito. So as malaria goes from being inside a human, it lives in the human bloodstream. It's intracellular. It's in a cell that's full of protein and sugar, and so it doesn't have a lot of resource limitations. And so as it moves into the mosquito, it's extracellular. It has to be motile. It's in a much more hostile environment. And so sort of hypothesized, and there isn't a lot of strong evidence towards this, and this is something we're trying to address, but what's hypothesized is that they ramp up mitochondrial function to be able to kind of survive in that more hostile environment and make better use of the limited resources that are there. And so that makes them extra vulnerable to inhibition of that organelle during that process. And so all of these drugs that we tested, something like, I think, 95% of the hits we got from a screen were mitochondrial inhibitors. And that kind of highlighted one that kind of demonstrated that fact that what we were seeing is that even in drugs that work both in the blood stages and in the mosquito, that there was a kind of differential extra inhibition potency in the mosquitoes. They were more vulnerable in the mosquito, but it also highlighted how little we actually know about what's important which mitochondrial functions are important. So the kind of main thrust of the lab as is, and we're still getting things off the ground, we're pretty new, but the idea is we're kind of focusing on this translational aspect of making new products like this anti-malarial treated bed net. That's one part of it. But the other is to try and really figure out what functions of the mitochondria are important specifically during mosquito development, so that we can perhaps identify other targets for mosquito-targeted inhibition, but also potentially new prophylaxis for the liver stages as well. So I mentioned that the mosquito stages of the parasite are hard to study, but you have to study those, or you have to be able to grow those to get to the liver stage, which is also kind of understudied as a result. It's just hard to get to the liver. There's a lot of research there because it's a vaccine target. But one thing we don't have as a kind of field is a lot of prophylactic treatments, or treatments that prevent the establishment of infections that are usable in endemic countries. So that's another area that we think maybe we can, by studying the mitochondria, which is a target of, as I mentioned, a tovacone, it's a prophylaxis, it can't be used in endemic settings because it generates resistance very, very, very quickly. So it's used in travelers. It's a great drug, or it's a potent drug, I should say. It's not a particularly good drug, as I mentioned, but it's not used in endemic settings because of the potential for resistance arising in blood stage infections. So when you give someone a prophylactic drug as a traveler, they're not carrying an infection in their blood.
Right.
But in an endemic setting, you can give them a prophylactic treatment, but they might be asymptomatically carrying malaria in the blood, and the resistances might emerge there. And so there's this kind of need for seasonal prophylaxis drugs in sub-Saharan Africa, and we think the mitochondrium might have other targets in it that would be useful, but it's basically a black box outside of the blood stages. And so we really want to kind of try and take the mitochondria into pieces and look at how it changes the way the parasite grows in the mosquito.
It's interesting that you characterized the parasite being in a more hostile environment when it's in the mosquito, because maybe it's just my bias, but when I think of host-parasite interactions, I guess I go to immune defenses, and I think of, even though I'm not an immunologist, I think of in the human host. So I'd never really considered that about the mosquito being a tougher environment.
Well, I mean, I think, there's tough and tough, right? So the mosquito is mounting an immune response too, and it's important. We know it's important for kind of controlling the it doesn't prevent infection. You're never going to get to sterilizing immunity in a mosquito, but it does kind of contain the infection to kind of manageable levels for the mosquito, if you knock out elements of the mosquito's immune response, which is only an innate immune response. They don't have any adaptive immune response. You can get fitness effects on the mosquito from infection. So normally the mosquito isn't affected by Plasmodium. If you give a kind of natural Plasmodium mosquito pairing, like Anopheles gambiae and an African Plasmodium falciparum, the mosquito is unaffected. It's happy. It just produces the same number of eggs, lives for the same amount of time, doesn't really seem to have any ill effects from that infection. But if you start messing with its immune response, you get negative deleterious effects on the mosquito. And so we think strictly the parasite isn't really a parasite of mosquitoes anymore. It's more of a commensal relationship. But yes, in the human, right, so when the parasite's outside of a red blood cell, it's in a hostile environment, it's under attack for the immune system, but it's hiding in a red blood cell most of the time. And it's in a bag full, and I have a former colleague, I'm a nose to your eye saying, who would kill me for saying this, but the red blood cell is just a bag full of glucose and protein. So, it's in terms of its kind of trophism, right? Its ability to grow and bring in nutrients. It's in a very cushy spot altogether, and it's, happily fermenting glucose instead of properly metabolizing it. So, and it doesn't matter about all the lactic acid it's producing because it's just,
That's the host problem.
Yeah, exactly.
Yeah.
It's just in this urine.
I feel like it has its own liquid IV, if you will.
Yeah, exactly. That's it.
Right in the RBC, so it's happy to be there, which is, fascinating. But yeah, I like the idea of targeting the mosquito, and I don't know that we know a ton about that, and I'm glad you're working on that. But one question I have is to kind of break it down back to the mitochondria. So can we talk about the mitochondria and what it's doing for the parasite and, why it's an important organelle?
Yeah. So that's the kind of running joke is the mitochondria is the engine of the cell, right? And
Right.
that's a gross simplification, right? And in fact, in the blood stages, it's exactly not what the parasite is using its mitochondria for. So we know, in fact, that a lot of parasites like Plasmodium and some of its more closely related apicomplexan parasites have got this very, very reduced mitochondrial genome and very reduced functionality. So they've lost some functions. They can't do beta oxidation, which is the process of turning fats into energy. So Plasmodium can't do that, it has to rely on sugar or amino acid. And it's lost other functions too. And in fact, in humans, in the human infection stages of Plasmodium, it doesn't really use its mitochondria to produce ATP at all, which is that kind of classic function from high school biology, right? The mitochondria is for producing ATP. And in fact, all it's doing is it's running through all those processes to produce redox potential, like electron carriers for one function, which is dihydroorotate dehydrogenase. Don't say that when you're drunk. And what that enzyme does is it produces nucleic acids. So it produces nucleic acids for DNA synthesis. And it's not even it's not part of any of the canonical processes in terms of the mitochondrial electron transport chain or TCA cycle. It's just taking advantage of all that electron flow to power its own function. And so that's the only known function that's essential to parasites in humans. However, we know from Cryptosporidium, which is a closely related parasite, that it's evolved very, very far down that parasitism road, and it no longer has a mitochondria. It has this kind of very reduced organelle called a mytosome instead. So it's lost all of those functions. And so in that case, you'd assume if Plasmodium was happy with just that single function, it would have lost some of these other functions. But it actually does have a canonical full TCA cycle and a full electron transport chain and maintains all of these enzymes. Apparently, as was thought, just for the function of this one enzyme. But what seems to be the case now is in the mosquito, you get this kind of activation of all these pathways. And we just don't know. So there's not just the energy production, the TCA cycle, but there's a load of biosynthetic pathways that come out of the mitochondrion as well. Heme biosynthesis, that's the kind of process by which you kind of combine iron into proteins for functional reasons. The pyridines I mentioned, the nucleic acids. I'm blanking on all the rest of them too. But there's a few. So, but primarily, I think the big function that we're interested in is actually ATP production, because it's something that's not important to the parasite at all during that human blood stages, but it is important. We know in the mosquito, I think at this stage, we don't actually have any evidence for that. Actually, we're working on it.
It's a hypothesis.
But yeah,
right?
our hypothesis is that it's so there's some signs that. So there's certainly more going on than just this DHODH, dihydroorotate dehydrogenase function, because we've used inhibitors for that drug against the mosquito stages, and they do nothing. So So we know atovaquone works, but for example, a DHODH inhibitor does not, and those two functions are actually really closely tied together, which suggests that it's more than just this pyrimidine synthesis that's important. So that's where we're kind of starting as ATP synthesis, but we're also interested in some of these other biosynthetic pathways as well, and how the parasite uses carbon sources. So, they can use amino acids, they can use glucose. We don't know which is more important. We think it's amino acids, but again, there's no evidence there. So we want to kind of parse out carbon metabolism and think about how ATP is produced and whether this target's there for both mosquito and liver stage intervention.
So one question that brings to mind for me is we have a switch over then from the parasite happily in a red cell with minimal mitochondrial function, and then ramping that up. What do we know about the control of mitochondrial function? Is it that in the human host, it's just not making some of these enzymes that are required in the mitochondria, and that expression's turned on in the mosquito, or is there something else?
Yeah, that seems to be the case. So yeah, it's actually really interesting because I always used to, prior to really getting into this, I used to, you just conceptualize mitochondria as being lots and lots of little organelles within a cell. Like in a human cell, you have multiple mitochondria, whereas Plasmodium has a single mitochondrion, singular, right? So, and it goes from this kind of little lozenge-shaped thing during the asexual cycle to this really highly incredible, I have a great picture actually, I will share it with you. A really highly branched organelle. It's all still a single organelle, but it's highly branched and kind of throughout the entire cell. And we don't really know what's driving that factor. Actually, some of our colleagues here at UGA, Diego Hewitt, is studying some of this as well in terms of how the mitochondrion is inherited during kind of division of the cell. and how it's subdivided. But it goes through these really massive dynamic changes. So it's in asexuals, again, in the blood stages, it doesn't have any cristae. This is this classic formation, this invagination on the inside of the mitochondrial inner membrane. That's actually really important for ATP synthesis. That's what it's for. It increases the efficiency and surface area of ATP synthesis. And what we know is that as the parasite gets ready to become infectious to a mosquito, it forms a special stage called the gametocyte, which is infectious to mosquitoes. You start seeing these invaginaceous forming if you look at the ultrastructure of the parasite. So the mitochondria is not only kind of becoming more important, it's growing, it's increasing its surface area to kind of be able to be more functional. And we do see increase of expression of those kind of energy production related genes as well. So
that process begins while it's still in the human.
Yeah, so it's getting ready for that.
Transmissible form, and then it's ready to hit the ground running in the mosquito.
That's right. Yep.
I have a really naive question. And I guess I'm thinking about it because we're always talking about malaria in the human host and how there's different stages of the parasite. But then I know when the parasite goes into the insect, it has to go from the gut to the saliva eventually, right? And then, So are there different stages of the parasite in the mosquito? And then I guess as a secondary question to that, are those stages differentially targetable?
That's a really, really great question. And yes, there's a lot of different stages. So I mentioned prior to infection, some proportion of the blood stage parasites will kind of become sexually active effectively, right? They'll reach adolescence, if you like. So I think some 1% to 2% of asexuals will kind of cycle through and become the sexual form. So you get male, and this is a eukaryotic organism like humans, and it has male and female gametes. And so it produces these, they're called gametocytes, male and female gametocytes that kind of hide in the bone marrow of the humans and develop over a couple of weeks into mature forms. And those then migrate to the peripheral circulation, so in your skin, so they can be picked up by mosquitoes. And that's the mosquito infectious form. So within a couple of literally minutes upon ingestion, those male and female gametocytes will activate because of the drop in temperature and an increase in pH. So the gut of a mosquito is alkaline. And so they detect that environmental change and kind of burst out of their host red blood cell. And the males actually undergo a process called exflagellation, which actually causes them, they look a lot like human sperm cells or mammalian sperm. And the females, it's very, very, very similar to an egg and a sperm, actually, which is kind of, it still blows my mind given how distantly related these organisms are from metazoan life, right? So this fertilization, and that's kind of a really important part, and actually what makes studying the parasite in the mosquito more challenging is that the parasite undergoes sexual reproduction inside the mosquito's midgut, inside that blood meal. And so they recombine genes, and for a period of time it has 2 copies of its genome, which makes things like knockout studies a bit more complicated. But yes, so there's a fertilization that forms a diploid zygote that then differentiates into an okinate, and that's the form that's motile, and it's responsible for kind of burrowing out of the midgut of the mosquito. So it burrows out of the midgut, forms a cyst on the outside of the gut. That cyst, which we call an oocyst, that will then grow for a period of about a week. It just goes through a lot of replication, and it's got, I think it will produce something like 5,000 to 6,000 daughter sporozoites, we call them. I wouldn't get too hung up on the technical terms. Parasitologists always come up with the wackiest names for the various different stages of their parasite. But yeah, so the oocyst produces sporozoites. Those are also motile, but they kind of get into the open circulation of the mosquito, and they end up in the salivary glands that way, and they can actively invade the salivary glands, and they sort of sit there waiting for the mosquito to take another blood meal. So, and that whole process takes 2 weeks in the lab anyway. It's probably, as I mentioned earlier, a little bit faster in the field, but because we don't usually feed our mosquitoes more than once.
So is that the stage that's most susceptible to these mitochondrial drugs?
Oh wait, yes. So yes, the as far as we can tell, the most susceptible stage to mitochondrial inhibition is the okinate, and that makes sense. It's in a very hostile environment in the midgut. It's also motile. It has to, and it's under time pressure in a way that sporozoites are. So let me see if I can make this make sense. So the mosquito, obviously sometimes we have to remind ourselves the mosquito is not taking a blood meal in order to be infected by malaria, right? It's taking a blood meal so it can get all that protein and lipid and turn it into eggs. And so, but it's also full of iron and other sorts of toxic stuff. And so she wants to get rid of that as quickly as possible. So it takes a mosquito somewhere from 48 to 72 hours to fully digest a blood meal. And then all of that kind of iron and other stuff that she doesn't want is excreted. And so the parasites are really on the clock to get out of the midgut. And so the fertilization process takes minutes, but the development of zygote to okinate takes about 21 to 24 hours. And if those O kinases don't get out of the midgut in time, they're just going to be excreted with the rest of the gut contents. And so they're really, really under a lot of time pressure. They have to be motile, and they're in a kind of life stage or an environment where they can't really gather resources because they're surrounded by digestive enzymes and reactive oxygen species and all sorts of horrible stuff. And so they're very vulnerable to inhibition. And in fact, the original paper that we published in 2019, that's where when we took mosquitoes and exposed them to a tovacillin through their legs, and then blood fed them, and then dissected them to see where the parasites were dying, it was O kinase where we were seeing them dying. So they're very, very vulnerable to that inhibition. And I think part of the reason it's most effective is because there's no chance for them to recover and kind of keep going. Mm-hmm. We found later that if you expose the later stages, the oocysts, they stop for 2 or 3 days. They stop growing, they're arrested. But then when the drug concentration goes below a certain threshold, they start growing normally again because they're still in a hurry because mosquitoes only live 3 weeks and parasites take 2 weeks to develop. So they're still on the clock, but not as much as an O kinase is.
Right.
So I think some of your earlier field work was in mosquito behavior. Is that correct?
Yeah.
How do we take advantage of known mosquito behaviors to deliver this, the drug you're working with or a new compound?
That's a really interesting question. So mosquitoes are very small and the environment is very big. And so you can't go out and individually treat every single mosquito. And in fact, that leads us to, for example, male Anopheles gambiae, we don't know where they are in the environment. They are distributed. They're under like a blade of grass or under a leaf or hiding in the eaves of your roof. But we don't know where they are, except when they're swarming, which is an aggregation behavior they use for mating. And that's what I was studying, actually. As it turns out, that's not particularly useful aggregation, but basically for control of mosquitoes, it's entirely dependent on aggregation behavior. So we know that females want to be around hosts. The most efficient vectors of malaria are all very, very human adapted. So they want to feed on humans particularly. And so we know that those mosquitoes are going to be aggregating around hosts. They all have different species have different windows of time when they want to feed. And Anopheles gambiae, again, which is this very, very important vector, likes to feed at night. And that's why bed nets work, because you sleep under a bed net and they're attracted to you. That's an aggregation behavior. And then insecticide kills those mosquitoes as they aggregate around a host source, right? And that's basically, in terms of taking advantage of that behavior, that's the behavior that all current mosquito intervention tools really take advantage of the most. Is either through spraying walls, so after a blood feed, the mosquitoes will often rest on the inside of walls, or through these treated bed nets. And for an anti-malarial approach, for us, I think the most likely initial application is through also using a bed net technology. It's very, very effective. It's highly proven tech. We have that aggregation behavior associated with it. And so it's a good way of treating a lot of mosquitoes in a short period of time. And so that's definitely one of the kind of most advantageous ways we can take advantage of that behavior. The other, as I mentioned, the other major aggregation behavior is swarming. So you get very few females, but you can get tens of thousands of males. We actually, at one point, were kind of playing with the idea of using drones to kind of mass treat, because the swarms happen in the same place every night. So that they're kind of very spatially predictable. And so theoretically, those suggested you could program drones to kind of go and spray swarms. When you model this out, most males don't mate in their lifetime. So when you have a swarm, I have seen and worked on swarms at 10,000, 15,000 males, and we maybe capture 15 or 16 mating couples coming out of the swarm. So most males are not successful, and most females get mated and they just go off on their way. And so actually reducing the success or individual male success is actually not a particularly effective way of targeting these things, although it's...
So in the dating game, the male mosquitoes are mostly losers.
Oh yeah, really losers.
I was just thinking I had a lot of things to say, but I'm just gonna stay silent here as the only female in the room.
Yeah.
Okay. So this has been fascinating, Doug. One of the things that we like to ask our guests before we end the podcast is, well, maybe not every guest, this question is very specific to you, but so when you're not thinking about malaria and mosquitoes, what are you doing in your free time?
So mostly trying to stop a 3-year-old from murdering himself. This has been my life. We have my son Alistair. He's hyperkinetic. He's very busy, and he's always trying to. But before that, when I had a life, I used to. I am a pretty enthusiastic road biker. I used to cycle quite a lot prior to becoming a dad. I do it much less now, and much less since moving to Athens. It's a little bit riskier than it is in Cambridge, Mass. But cycling is definitely my big time sink when I'm outside of work, for sure. It's a really great way of freeing up the language centers of your brain to work through some problems while you're not really concentrating on anything else is great.
So our guest today is Dr. Doug Paton, faculty in the Department of Infectious Disease and member of the Center for Tropical and Emerging Global Diseases. Doug, thanks for being with us.
It was a great pleasure. Thank you.
Links to additional information about Dr. Paton's research can be found on our Instagram account, which is @PPPPodcastUGA.
As always, we appreciate you tuning in today. And if this is your first time listening to the People, Parasites, and Plagues podcast, we encourage you to check out the other exciting episodes launched last season. Please rate the podcast, and if you have any questions or suggestions for future episodes, reach out to us via email at ppp@uga.edu. This podcast is brought to you by the Faculty of Infectious Diseases and the Grady College of Journalism at the University of Georgia. It is supported by the University of Georgia through the Office of Research, the UGA Graduate School, and the College of Veterinary Medicine. Special thanks to our production assistant, Sid Wigand, for research, editing, scheduling, and keeping us on track.
Ta-da!
Ta-da!
Fantastic.